Hi students, today we are going to discuss about very important research in biological science, that is Human Genome Project, that is also known as HGP. So what do you mean by genome? So genome is the whole genetic content of our organism, of our body. That is the genomics. Genomics is the field which deals with genome. So the sequencing, what do you mean by sequencing genome? Sequencing genome means it finds out the arrangement of nucleotide bases in gene. That is nucleotide bases means ATGC arrangement, adenine, thymine, guanine, cytosine. These are the nucleotide in DNA. So in the sequencing genome means scientists find out the arrangement of ATGC in whole genome. So actually this idea is started in 1984 by US government, and later the idea that the projects launched in 1990 by US government with the help of two organizations, that is US Department of Energy and NIH. NIH is National Institute of Health. And later some countries are involved in this project, that is Wellcome Trust in UK is the main partner of Human Genome Project, and also Japan, France, Germany and China also included or also being part of this project. And this Human Genome Project is also known as a mega project. So why this is called mega projects? Maybe because of some estimation. So let's see what are the estimations in Human Genome Project. The first estimation is the estimated to 3 into 10 raised to 9 base pairs to be sequenced. That is, the scientists assumed our genome contains 3 into 10 raised to 9 base pairs. So the estimated to be sequenced 3 into 10 raised to 9 base pairs. And the total cost of the Human Genome Project estimated to be $9 billion, and also the time needed to complete this project is 13 years. So that is the project launched in 1990, so it is expected to complete it in 2003, but in 2000, the HGP published the first draft of human genome. So in 2000, HGP published first draft of human genome, and the final draft is published in April 14, 2003. So that April 14, 2003 is a special day in biology, that is the 50th anniversary of publication of data that describing double helical structure of DNA by Watson and Crick. So that is the day April 14. In that day the final draft of the Human Genome Project is published. So these are the basic estimation of Human Genome Project, that is sequence to be estimated 3 into 10 raised to 9 base pair, and the total cost is $9 million, and the year completed need 13 years. So next, during that time there is no particular software for analyzing this big data. So the scientists are planning to develop some softwares for storing and analyzing data. So simultaneously a new branch in science evolved with Human Genome Project, that is bioinformatics. So bioinformatics is a new field of science that is evolved simultaneously with Human Genome Project. That is, say bioinformatics is a science, this is a interdisciplinary science which combine computer and biology. That is, this bioinformatics field uses computer programs for analyzing large amount of data. Also they created biological databases for storing this data. So the parallel field emerged with Human Genome Project is bioinformatics. And with the Human Genome Project, a special committee is evolved that maintain the ethical, legal and social issues about Human Genome Project. So, if there is any problem pop up with human ethical, legal or social issues, this committee will resolve. So, these are the basic idea of Human Genome Project. So, one interesting thing about this Human Genome Project is, with Human Genome Project that is maintained by the US government, and parallelly a private sector, that is Celera Genomics is a private sector in US that is owned by Craig Venter and launched in 1998. Owned by Craig Venter and it is launched in 1998. They parallelly sequence human genome with whole genome shotgun sequencing method. So, the Celera Genomics parallelly sequence Human Genome Project with US government. So, this Celera Genomics is compete with US government Human Genome Project. The main competition is for gaining patent over human genome. So, but in March 2000, the US President Bill Clinton announced that the Human Genome Project could not be patented and this should be made freely available for all researchers. So, the competition is over and in the June 2000, the first draft of Human Genome Project is published with Collins and Venter. So, the Collins is the person in the public US government NIH and the Venter is the person behind Celera Genome. So, both combined published the first draft of Human Genome Project in June 2000. So, this is the introduction of Human Genome Project. So, do not forget that it is launched in 1990. US Department of Energy and NIH is the two main organization of Human Genome Project and expected to be sequenced 3 into 10 raised to 9 base pairs. Cost is $9 billion and the year time needed 13 years. Actually, the time needed 13 years, that means the project started in 19, so expected to end in 2003. So, the final, as per estimation, the final draft is published in April 14, 2003 and the parallel field arise with Human Genome Project is bioinformatics and special committee is there for maintaining ethical legal issues. And do not forget, never forget the Celera Genomics and Craig Venter is one of the eminent scientists in the genomics and the whole genome shotgun sequencing is the one of the very important sequencing method which are used in genome sequencing. This shotgun sequencing genome sequencing is applied in various fields. Let us see what are the goals of Human Genome Project, that is what the scientists find out after Human Genome Project. The first one is human genome contains 3164.7 million base pairs, that is 3.6 billion base pairs. That is the size of human genome is 3.16 billion base pairs. That is if we write the human genome in a books, we need 3,300 books. That book, each book contain 1000 pages and each page contain 100 type letters. That is the size of human genome. That is, if we want to write the human genome in a book, we need 3,300 books. And this the size, this human genome, this base pairs is find out from the 24 chromosomes. 24 chromosomes means it is the amount, it is the sequence in haploid set of chromosome. We know the human contains 23 pair of chromosomes, that is 22 autosomes plus 1 sex chromosome. For the female, that is X chromosome, and in the case of male, the sex chromosome is Y. So, the scientists sequence this. the 22 autosomes plus X chromosome and Y chromosome. So, the 3.16 billion base pair is the size of 24 chromosome. And the second goal is the average gene in the human genome is 3000 bases. Only 3000 bases, the average gene, the size of the average gene is 3000 bases. The largest gene is dystrophin, which contain 2.4 million base pairs, and smallest one is TDF, that is testis determined factor, that is a Hollandric gene. Hollandric gene means genes present in the Y chromosome and that has 14 base pairs long. So, these are the largest gene and the smallest gene. So, the average gene size in the human genome is 3000 bases. And the next goal is the total number of gene is 30,000. So, total number of gene in human genome is 30,000. Actually, the scientists expected more numbers, but this is less than the expected ratio. So, the next is the function. The functions of the 50% of genes in the human genome is still unknown. So, this is 50% of gene function is still unknown in human genome. So, that genes are maybe non-coding genes. And the next goal is repeated sequence make up a large portion in the human genome project. So, repeated sequence means some nucleotides are repeated in the genome with different numbers and different individuals, and this repeated sequence are considered as unique identifiers, and that unique identifiers are used in DNA fingerprinting. So, most of the large portion of the human genome is made up with repeated sequences. And next goal is less than 2% of genome codes for protein. That is, the coding region is only 2%. So, the DNA which codes to protein is known as coding region. So, the amount in the coding region is only 2% in human genome. And the next goal is 1 million locations have SNP. So, what do you mean by SNP? SNP is single nucleotide polymorphism. So, polymorphism means multiple forms of genes. So, here SNP means polymorphism due to single nucleotide. That is ATGC is one gene sequence. Then the change in the second section, this T is become A. So, this is SNP and this causes some changes in the function of gene. So, this is known as single nucleotide polymorphism, change in one nucleotide that causes polymorphism. So, 1.4 million locations in the human genome project contain SNPs. And the next call is the largest chromosome sequenced in the human genome project is chromosome 1. That is, that chromosome 1 contains 2968 genes. So, largest chromosome in the human genome project is chromosome 1 that contains 2968 genes, and the smallest one is chromosome Y that contains only 231 genes. Actually, the full draft of human genome project is published in 2003, but the chromosome 1 is not completed on that year. The chromosome, because of the larger size, the chromosome 1 is completed in 2006. Okay. So, next the second goal of the ninth goal of the human genome project is the informations in the human genome project is stored in the biological database. The first I said the bioinformatics is a field that parallelly evolved with human genome project. So, in using bioinformatics we created biological database, and then the information of this genome is stored in different biological database. Also with bioinformatics, scientists develop various tools, biological tools for analyzing biological data. That is the goal of Human Genome Project. And besides that, the technology which are used in Human Genome Project, is also transferred to another sectors. Okay. The technology which used in the human genome can be applied to different sectors in the biological field. So, these are the goal, that is the findings, findings we get after Human Genome Project. So, don't forget the size of the Human Genome Project, that is human genome, that is 3164.7 million base pairs or 3.16 billion base pairs, and the average gene is 3000 bases, and less than 2% genome codes for protein, and 1.14 million SNPs are there, and the largest chromosome is chromosome 1 and smallest is chromosome Y. Next, let us see what are the methodology which are used in the Human Genome Project. Actually, the scientists assign two approaches, that is, one is based on sequence coding, so sequencing only coding sequence, and another one is based on sequencing coding and non-coding sequence. So, some scientists said why we go for non-coding sequence, but some scientists supported non-coding sequence. So, two approaches are arise. One is the sequencing coding region, that is EST. They are considering EST, that is expressed sequence tag. Expressed sequence tags. So, that is the sequencing coding region, and the non-coding region, sequencing coding and non-coding region is known as sequence annotation. So, this method is followed in Human Genome Project. So, they are sequencing coding region and non-coding region. For this, the entire DNA is extracted from the genome and cut into small fragments. This is the first step, extract the full DNA from the genome and cut into small fragments with the help of endonuclease. And these fragments are sequenced with the help of Sanger's DNA sequencing method. So, after cut into fragments, these small fragments of DNA are sequenced with Sanger's DNA sequencing method. And before going to the sequencing, these fragments are cloned. So, for cloning for cloning DNA fragments, we need host cells and vectors. In Human Genome Project, the host cell used is bacteria and yeast. So, bacteria is for prokaryotic and the yeast is for eukaryotes. And the vector which are used in the Human Genome Project is BAC and YAC. So, BAC means bacterial artificial chromosome. Bacterial artificial chromosome, and the YAC is yeast artificial chromosome, and in the Human Genome Project the vector used is YAC. So, yeast artificial chromosome is the vector which are used for cloning in Human Genome Project. And during the cloning, the sequence, the fragments are multiplied, and then this sequence are going to Sanger's sequencing method, and we find out the order of the nucleotide bases, and after that these sequences are assembled based on overlapping region. So, after sequencing, the small fragments of DNA is assembled by overlapping region. So, the generation of overlapping regions during fragmentation is very important in Human Genome Project. So, generation of overlapping regions. So, what is overlapping regions? For example, this is the sequence, and we are generating multiple copies of this, and cut into small fragments with overlapping regions. So, overlapping regions means we first cut AAT, GCC and CC, and in the next we cut ATG and CCC, CC. So, this region is the overlapping region. So, using this we can assign DNA sequences. So, that method is used in assembly method. So, for assembling DNA fragments we are using very highly efficient computer softwares like assemblers. We are using highly efficient computer softwares, assemblers for assigning fragments. So, this is the main methodologies. Actually, this methodology is the technique which are used in rDNA technology. So, using that we sequence small DNA fragment and then assemble this DNA fragment through overlapping regions by assemblers, by computer programs. After annotating the sequence, the genome sequence or the gene sequence is fragmented, is assigned to chromosomes. Okay, so two approaches, sequence coding and sequence non-coding. We follow the sequence annotation, that is we coding region and non-coding region. For this we extract DNA and cut into fragments and sequence those fragments using Sanger's method. After sequencing, we assemble this sequence using computer programs, assemblers, and then this assigned to, after annotation, this assigned to chromosomes. And for cloning the fragments we are using host and vectors. The host is bacteria or yeast, and the vectors we are used in the Human Genome Project is YAC, that is yeast artificial chromosome. So, next let us see what are the applications of Human Genome Project. So, these are the applications of Human Genome Project. The first one, it helps to diagnose disease and treatment. That is, there are so many diseases that is related to genetic disorder or that is related to gene sequences. So, this Human Genome Project helps to identify the main cause of these diseases, genetic diseases, and the doctors can easily assign the treatment for these genetic disorders, like genetic disorder like carcinogenic diseases and some metabolic disorders, all are related to gene sequence. So, using human genome, using the information provided from the human genome, we can easily identify the cause of genetic disorders and easily we can design the treatment for this genetic disorders. And the second one is treat diseases using rDNA technology. Actually, the rDNA technology is basically work with the knowledge which are provided from the Human Genome Project. For example, in the case of insulin. So, we know this is used for diabetics, and if we know the sequence of insulin, we multiply this insulin into with the help of E. coli and produces large amount of insulin, and that is used for treatment. So, the multiplication of insulin in the host cell with the help of rDNA technology. So, using rDNA technology we multiply this insulin sequence. So, for that we need the sequence information of insulin. So, for treating some diseases, the Human Genome Project is used. And the next application is that the related technology, the technology which are used in the Human Genome Project is applied to non-human genome. That is, for example, bacteria, Drosophila, yeast, in the case of plants, rice. That is, the same technology is used for sequencing bacteria, Drosophila, yeast and rice. The first sequenced organism is Haemophilus influenzae. This is the first sequenced organism. So, the same technology, the technology used in the Human Genome Project is used for sequencing non-human genome. So, because of that we can improve and we can use this genome information to improve the agriculture field and also in the microbiology field, that is in the field which are using microbes as human welfare, and also in the field which microbes are used for energy production. So, these all fields are using the informations which are providing from genome projects. So, these are the main application of Human Genome Project. So, it helps to diagnose disease and treatment, and it is used in RDNA technology, and also related technology is applied in non-human genome or applied in various sectors. So, this is the idea of Human Genome Project. Actually, this Human Genome Project, we can say the Human Genome is the Google server of all Google server for genetic information of humans. So, humans genetic information, we can search any genetic information from human genome. So, this is like a Google server. So, this is the Human Genome Project, and don't forget, this is the most important research in the biological field. So, hope you understand this. So, in the next section we will discuss another topic related to genomics. Thank you.